Here, for important groups of immune cells that play major roles in protecting our body from infection and maintaining immune responses will be discussed. These are dendritic cells, macrophages, mast cells, and granulocytes. Let's begin with dendritic cells. Dendritic cells are known as the professional antigen-presenting cells of the immune system. Their primary function is to capture invading microorganisms or foreign antigens, process them, and present these antigens on their surface to activate T lymphocytes. In this way, they serve as a bridge between the innate and adaptive immune systems. Most dendritic cells arise from myeloid progenitor cells in the bone marrow. In healthy skin, there are three major populations of dendritic cells: Langerhans cells in the epidermis, conventional or myeloid dendritic cells in the dermis, and plasmacytoid dendritic cells. Conventional dendritic cells are abundant in barrier tissues such as the skin. Under normal conditions, they remain in an immature state, allowing them to efficiently capture antigens through phagocytosis using complement receptors and Fc receptors. After antigen uptake, they process the antigen and present it through MHC class II molecules to activate CD4-positive helper T cells. If the antigen enters the cytoplasm, it is processed by the proteasome and presented on MHC class I molecules, activating CD8-positive cytotoxic T cells. They are also capable of cross-presentation, an important mechanism for eliminating virus-infected cells and maintaining immune tolerance. Different dendritic cell subsets can be identified by their surface markers. CD141 positive and CD1C positive cells represent conventional myeloid dendritic cells. Monocyte-derived dendritic cells express CD14, while plasmacytoid dendritic cells express CD123 and CD303. Langerhans cells express CD1A and Lang-Aron, also known as CD207. Plasmacytoid dendritic cells. These cells specialize in antiviral defense. They express toll-like receptors 7 and 9, which recognize viral RNA and DNA, allowing them to detect viral infections very early. Once activated, plasmacytoid dendritic cells produce extremely large amounts of type I interferons, nearly 10,000 times more than any other cell type. Although they are less efficient at antigen presentation because they express lower levels of MHC class II and co-stimulatory molecules, their interferon production is essential in controlling viral infections. Clinically, plasmacytoid dendritic cells contribute to the development of psoriasis. In this disease, the antimicrobial peptide LL37 binds to self-DNA and activates TLR9, resulting in excessive immune activation and skin inflammation. Langerhans cells. Langerhans cells are specialized dendritic cells located within the epidermis, where they make up approximately 2 to 4% of epidermal cells. They are positioned between keratinocytes and continuously monitor the skin for invading pathogens. A unique feature of Langerhans cells is the presence of Birbeck granules, which have a characteristic tennis racket appearance under the electron microscope. They express high levels of CD1A and CD207 or Lang-Aron, which helps internalize antigens and promote cross-presentation through MHC class I molecules, allowing activation of CD8-positive T cells. Macrophages. Macrophages are essential components of innate immunity and inflammation. They act as tissue-resident immune sentinels, constantly monitoring tissues for infection and injury using scavenger receptors, pattern recognition receptors, and phagocytic receptors. In healthy tissues, macrophages maintain themselves with minimal contribution from circulating monocytes. However, during infection or tissue injury, monocytes migrate into tissues and differentiate into macrophages or dendritic cell-like cells. Macrophages are classified into two major functional types. The first type is the M1 macrophage, also known as the classically activated macrophage. These cells are activated primarily by interferon gamma. They produce pro-inflammatory cytokines including IL-1 beta, TNF-alpha, IL-12, and IL-18. They express high levels of MHC class II, CD68, CD80, and CD86. M1 macrophages are highly efficient at phagocytosis and killing intracellular bacteria. Interestingly, vitamin D enhances their ability to eliminate intracellular pathogens. The second type is the M2 macrophage, also known as the alternatively activated macrophage. These cells are activated by IL-4, IL-10, IL-13, and TGF-beta. Instead of promoting inflammation, M2 macrophages are involved in tissue repair, wound healing, parasite defense, allergic responses, and resolution of inflammation. Several important receptors are associated with macrophages and antigen-presenting cells. CD40, CD80, and CD86 provide co-stimulatory signals necessary for T-cell activation. CD25 and CD127 are receptors for IL-2 and IL-7, respectively. CD206, also called the mannose receptor, recognizes microbial carbohydrates, while CD302 participates in cell adhesion and phagocytosis. Dectin-1 recognizes fungal glucans and contributes to antifungal immunity. Mast cells. Mast cell. Mast cells are abundant in the skin and mucosal tissues, where they serve as one of the body's first lines of defense. They originate from bone marrow precursors but mature only after entering tissues. These cells are long-lived and can survive for months to years. Mast cells may be activated through several mechanisms. The classic pathway is IgE-mediated activation, responsible for immediate allergic reactions. They can also be activated by IgG antibodies, pattern recognition receptors, complement proteins C3a and C5a, neuropeptides such as substance P, and toxins from bacteria or venom. When activated, mast cells rapidly release preformed granules containing histamine, serotonin, TNF-alpha, VEGF, proteases, cathelicidins, and chemokines. They also synthesize additional mediators such as IL-3, IL-4, IL-5, IL-6, leukotrienes, prostaglandins, and platelet activating factor. These mediators increase vascular permeability, causing edema and producing the classic wheel and flare reaction seen in allergic responses. Mast cells also recruit dendritic cells and T cells, enhance antigen presentation, and promote Th2 immune responses. Clinically, mast cells are responsible for conditions such as urticaria, commonly known as hives, and anaphylaxis, a severe life-threatening allergic reaction. Granulocytes. Granulocytes are white blood cells characterized by lobulated nuclei and cytoplasmic granules. They include neutrophils, eosinophils, and basophils. Neutrophils are the most abundant circulating white blood cells and serve as the body's primary defense against bacterial infections. They eliminate pathogens through phagocytosis, degranulation, production of reactive oxygen species, and formation of neutrophil extracellular traps, or nets. Nets consist of DNA coated with antimicrobial proteins such as myeloperoxidase and elastase. These structures trap microorganisms, prevent their spread, and facilitate their destruction. Neutrophil granules also contain antimicrobial substances including lactoferrin, lysozyme, cathepsins, and defensins, all of which contribute to pathogen killing. Basophils are important in allergic reactions and defense against parasites. They produce large amounts of interleukin-4, promoting Th2 immune responses. Eosinophils are particularly effective against parasites and are commonly involved in allergic diseases. They are recruited into tissues by IL-5 and chemokines such as eotaxins. Eosinophils interact closely with mast cells, and together they amplify allergic inflammation. In conclusion, dendritic cells, macrophages, mast cells, and granulocytes each play unique but complementary roles in the immune system. Dendritic cells initiate adaptive immunity through antigen presentation. Macrophages eliminate pathogens while regulating inflammation and tissue repair. Mast cells provide rapid defense and mediate allergic reactions. Granulocytes, particularly neutrophils, eosinophils, and basophils, are powerful effector cells that destroy pathogens and coordinate inflammatory responses. Understanding the functions of these immune cells helps us better appreciate how the body defends itself against infections while maintaining immune balance.